In 2019, a team of researchers published the complete genome of the Komodo dragon, Varanus komodoensis, the first full genome sequenced for any member of the family Varanidae, and one of the most information-dense genomes yet produced for any reptile. The Komodo dragon is the largest living lizard on Earth, reaching 3 meters in length and over 70 kilograms, capable of killing prey as large as water buffalo and pigs many times its own body weight. The genome was sequenced to understand the biological basis of those capabilities, the cardiovascular adaptations that allow a reptile to sustain the kind of burst activity that should be physiologically impossible for an ectotherm, the chemosensory systems that give it a detection range measured in kilometers, the immune adaptations that protect it in an environment saturated with bacteria from rotting carcasses. All of those questions were answered, but the most significant result the genome produced was phylogenetic. It confirmed, at a resolution that earlier molecular studies had approached but never achieved, exactly where monitor lizards sit in the reptile family tree. The answer had been suggested by previous work going back to the early 2000s, and it had become increasingly well supported as more squamate DNA became available. The genome made it definitive. The closest living relatives of all monitor lizards, of every species in the genus Varanus, from the tiny Varanus brevicauda of Australia at 20 centimeters to the Komodo dragon at 3 meters, are the snakes. The genus Varanus is one of the most ecologically successful lizard lineages in the history of reptiles. It currently contains around 80 recognized species distributed across Africa, Southern Asia, and Australia, with the greatest diversity concentrated in Australia, where roughly half of all known varanid species are found. Varanids first appear in the fossil record in the late Cretaceous, approximately 80 million years ago, in what is now Central Asia. From that origin, they dispersed across the Old World and eventually reached Australia, probably crossing through Southeast Asia during periods of lower sea levels when the Sunda Shelf was exposed as dry land, where they underwent an extraordinary adaptive radiation. The Australian varanids range from insectivores barely larger than a gecko to apex predators capable of taking kangaroos. They occupy deserts, rainforests, wetlands, coastlines, and everything in between. What unites them across all of this diversity is a suite of shared anatomical and physiological features that separate them from every other lizard family. The forked tongue used for chemosensory sampling, the active and often pursuit-based hunting strategy, the relatively large brain, the high aerobic capacity compared to other lizards, and, as genomic work has increasingly made clear, the venom system. The varanid body plan has proven extraordinarily adaptable. Individual species have independently evolved semi-arboreal habits, semi-aquatic habits, marine foraging, dietary specialization for eggs, dietary specialization for mollusks, and body sizes spanning 2 orders of magnitude. A lineage that can produce both a 20-centimeter insectivore and a 3-meter apex predator from the same basic genetic toolkit across the same geological epoch is a lineage that has found a profoundly flexible evolutionary strategy. The relationship between monitor lizards and snakes is not immediately obvious from their appearance. A Komodo dragon and a reticulated python look like they belong to different categories of animal. One is broad and heavy-bodied, with 4 powerful legs, and moves with a deliberate terrestrial gait. The other is legless, moves by lateral undulation, and has a body plan so different that it was once thought to represent a completely independent evolutionary experiment in vertebrate locomotion. But the molecular data tells a different story. Squamate reptiles, the group containing all lizards and snakes, have been studied with progressively more complete genomic data since the early 2000s, and the picture that has consistently emerged is that snakes are not a separate evolutionary lineage that evolved alongside lizards. Snakes are lizards. They are the descendants of a lizard ancestor that lost its legs and reorganized its body plan for a different style of locomotion. And the lizard group most closely related to snakes, the group from which snakes most recently diverged, is Varanoidea, the superfamily that contains monitor lizards and their immediate relatives. A 2013 phylogenomic study by Pyron and colleagues that analyzed DNA from over 4,000 squamate species confirmed this relationship with high confidence. The 2019 Komodo genome added chromosomal and structural genomic evidence that put the question beyond serious dispute. Once you know this, certain features of monitor lizards that seemed like superficial quirks become something else entirely. The forked tongue that both monitors and snakes use for chemosensory sampling is not a coincidence or a convergence. It is shared ancestry. The elongated neck and the way the head moves independently of the body is shared ancestry. The general sensory orientation toward chemical detection, rather than color vision, is shared ancestry. The animals look different because one lineage kept legs and the other did not. But the underlying architecture is the same because the lineages share a common ancestor that no other living squamate shares with them. The full evolutionary implication of the monitor-snake relationship becomes clearest when you look at a grouping called toxicophora. Toxicophora is a clade of squamate reptiles that includes snakes, monitor lizards, iguanas, chameleons, and several other lizard groups. Roughly 2/3 of all living squamate species. The name means venom-bearing, and it was proposed by herpetologist Brian Fry and colleagues in a 2005 paper in Nature that made a claim most of the scientific community initially found difficult to accept. Venom had not evolved independently multiple times in different snake and lizard lineages. It had evolved once in the common ancestor of all toxicophora and had been retained in modified or vestigial form across the entire clade ever since. The evidence for this was biochemical. Fry's team identified homologous venom gland proteins across an extraordinary range of squamates, including species that had never been considered venomous. Monitor lizards showed venom proteins in oral gland tissue. Iguanas showed them. Beaded lizards showed them. Species traditionally described as harmless turned out to have functional or ancestral venom systems. The venom that allows cobras to kill and that allows some colubrid snakes to immobilize prey had not evolved independently dozens of times across squamates. It was an ancient inheritance, modified in different directions by different lineages over 200 million years. In some lineages, the venom system became a primary predatory weapon. In others, it was retained as a low-level biochemical system with limited delivery apparatus. In a few lineages, it may have been completely lost. But the shared molecular architecture of the system, the same protein families, the same gland homologs, the same basic biochemistry, is the signature of a single evolutionary origin, not of convergence. The toxicophora hypothesis restructured how the entire history of squamate predation is understood, and monitor lizards sit at its center. The most famous application of this venom research was to the Komodo dragon, and it overturned one of the most persistent myths in popular herpetology. The standard explanation for how Komodo dragons kill large prey had been, since at least the 1980s, that the bacteria in their saliva was the operative mechanism. The story was that Komodo dragons would bite prey, and the bacteria-laden wound would become septic over days or weeks, eventually killing the animal while the dragon tracked it. The bacteria theory was repeated in textbooks, documentaries, and wildlife programs for decades. Brian Fry's 2009 study in PNAS dismantled it. Using MRI imaging of Komodo dragon skulls, Fry's team identified mandibular glands, venom glands, that were distinct from salivary glands and that contained a mixture of anticoagulant compounds, compounds that lower blood pressure, and compounds that induce shock. The mechanism by which Komodo dragons kill large prey is venom-assisted hemorrhagic shock, not bacterial sepsis. The bacteria in Komodo saliva are no more concentrated than in any other large carnivore that feeds on carcasses. The killing mechanism is the venom. The bacteria story was wrong. The 2019 Komodo genome, produced by Lind and colleagues at the Gladstone Institute, addressed the physiological question that the venom work had left open. How does a reptile sustain the activity level required to be an effective apex predator? Reptiles are ectotherms. They regulate body temperature from external sources rather than metabolic heat. And conventional understanding held that this imposed strict limits on sustained aerobic activity. Komodo dragons violate those limits in ways that had puzzled physiologists. The genome provided molecular explanations. Gene families associated with cardiac function, specifically genes governing calcium ion transport in heart muscle cells, showed signatures of positive selection in the Komodo dragon lineage that are otherwise seen in mammals and birds, not in reptiles. The modifications allow the Komodo dragon's heart to function with an efficiency closer to an endotherm's than a typical ectotherm's, enabling higher sustained output during activity. Genes associated with mitochondrial function similarly showed expansions consistent with higher aerobic capacity. The genome did not prove that Komodo dragons are warm-blooded in any conventional sense. What it showed was that the boundary between ectotherm and endotherm physiology is not as sharp as textbooks have suggested, and that at least one reptile lineage has evolved cardiovascular genetics that push substantially toward the endotherm end of that spectrum. Beyond the physiology, the 2019 genome also characterized the chemosensory system that makes monitor lizards such effective hunters at long range. Monitor lizards use their forked tongue to collect chemical particles from the air and deliver them to the Jacobson's organ, a paired chemoreceptor structure in the roof of the mouth, in what amounts to a biological chemical analysis system. The Komodo dragon genome showed expansions in gene families encoding olfactory receptors and Jacobson's organ receptor proteins that exceed what is seen in other reptile genomes, providing a molecular basis for the observed ability of Komodo dragons to detect carrion from distances of up to 10 kilometers under the right conditions. The forked tongue is not decorative. The two tines of the fork allow the animal to sample chemical gradients simultaneously from 2 points in space, providing directional information about the source of a scent. Essentially, a biological stereo system for chemosensory navigation. The genome showed that the molecular infrastructure underlying this system is deeply shared between monitor lizards and snakes, consistent with their shared ancestry, and substantially more developed in both groups than in other squamates. The extinct relatives of monitor lizards extend the story of the group's diversity into geological deep time. Mosasaurs, the enormous marine reptiles that dominated the world's oceans during the late Cretaceous, reaching lengths of over 17 meters in the largest species, have been placed by comparative morphology and molecular clock analysis as close relatives of varanoid lizards, the group that contains monitor lizards and their nearest relatives. Ancient DNA cannot be recovered from Cretaceous material. The chemical half-life of DNA makes recovery impossible beyond approximately 1 million years under even optimal preservation conditions. so the mosasaur relationship cannot be established through direct genomic comparison. But the anatomical evidence is extensive, and molecular clock estimates of when the varanoid lineage diverged from its nearest relatives are consistent with the mosasaur fossil record. The implication is that the lineage that eventually produced monitor lizards also, at some point in the Cretaceous, produced a radiation of marine predators that were among the largest and most successful apex predators in ocean history. When the asteroid impact that ended the Cretaceous eliminated mosasaurs along with non-avian dinosaurs, the terrestrial varanoid lineage continued, and the ancestors of modern monitor lizards inherited a world emptied of many of their competitors. The largest monitor lizard known from the fossil record is Varanus priscus, commonly called Megalania, which inhabited Australia from approximately 1.6 million years ago until its extinction roughly 40,000 years ago. Size estimates for Megalania have varied substantially over the decades, partly because the fossil material is fragmentary and scaling from lizard bone dimensions to total body length is complicated by the fact that large varanids are not simply scaled-up small varanids. Their body proportions change allometrically with size. Current estimates, based on the most complete material, suggest a maximum length of around 5 to 7 meters and a body mass of up to 600 kilograms, making it the largest terrestrial lizard in the Cenozoic record and a plausible candidate for the largest venomous animal ever to have existed, if the venom systems seen in living varanids were present in Megalania, which, given the deep conservation of those systems across the family, they almost certainly were. Megalania was a contemporary of early humans in Australia. The first Australians, arriving from Southeast Asia somewhere between 65,000 and 50,000 years ago, would have encountered an animal capable of taking prey the size of a large wombat or a juvenile diprotodon. The timing of Megalania's extinction, approximately 40,000 years ago, coincides closely with the broader megafauna extinction event that eliminated most of Australia's large animals in the same period. Whether Megalania was directly hunted, whether its prey base was depleted by human hunting, or whether climate change played a contributing role remains debated. But the timing fits the pattern seen in megafauna extinctions on every other continent where humans arrived during the Pleistocene. A lineage that had survived millions of years of climate fluctuation disappeared within a few thousand years of the first Homo sapiens. Monitor lizard intelligence has attracted research attention for decades, partly because their behavior in the field and in captivity consistently suggests cognitive capacities unusual for reptiles. Several varanid species have demonstrated the ability to count in controlled experimental settings, distinguishing between groups of prey items up to 6, a capacity that requires some form of numerical representation. Studies of monitor lizard foraging behavior have documented what appears to be cooperative hunting in some species, with individuals coordinating to separate prey from defensive positions. Learning experiments have shown that monitors can solve novel problems, form associations between previously unrelated stimuli, and retain learned behaviors across periods of months. The Komodo dragon genome provided a neuroanatomical basis for some of these observations. Brain-associated gene families showed expansions relative to other reptiles, particularly in regions associated with learning and memory processing. The monitor lizard brain is not structured like a mammalian brain, and direct comparisons of cognitive capacity are methodologically fraught, but the behavioral and genomic evidence together suggest that the varanid lineage has been under selection for enhanced cognitive function in a way that most lizard lineages have not. Pull all of this together, and the monitor lizard that emerges from the genomic and paleontological record is something very different from its popular image as simply a large, scaly predator. Varanids are the closest living relatives of snakes. Not a similar type of animal, but a genuinely close evolutionary sister group, sharing a common ancestor that no other living lizard lineage shares with them. They carry venom glands whose proteins are homologous to those in venomous snakes, inherited from a common ancestor that evolved the basic venom system over 200 million years ago. They have cardiovascular and chemosensory genetics that push the boundaries of what has been considered possible for reptile physiology. Their lineage in the Cretaceous produced the mosasaurs, the largest marine predators of their era. In Australia, they grew to the size of a large crocodile and coexisted with humans until human presence made that coexistence impossible. The 2019 Komodo genome was the first full molecular read of this history, and what it showed was a lineage that had been misunderstood at almost every level, misclassified in relation to snakes, mischaracterized in how it kills prey, underestimated in its physiological capabilities, and underappreciated in its evolutionary significance. The genome did not change what monitor lizards are. It changed what we understand them to have always been. Subscribe. New discoveries about lost species posted every few days.